Good afternoon. Looks like it's just the five of us for now, but it is 12:30, so we're gonna go ahead and get started. We have quite a busy schedule today. We have to finish the rest of lecture 3, and we will also start on lecture 4. So let's start with a bit of a recap of where we were last class. We were talking about the structure of a cell and the various organelles that you will find inside the eukaryotic cell. And we were discussing the endomembrane system specifically. The endomembrane system includes the nuclear envelope, which of course surrounds the nucleus, and that envelope extends into the cytoplasm to form a network of tunnels. We call those the endoplasmic reticulum. Some endoplasmic reticulum has ribosome on them, which makes them rough ER. Others don't have the ribosomes, and we call them smooth ER. The ribosomes are these tiny, tiny structures that are made up of proteins as well as RNA, and they are produced from the nucleolus, which is what you will find in the center of the nucleus. So once you make the ribosomes, they will exit through the nucleus, through the nuclear pore, and some will be freely floating in the cytoplasm. Others will be attached to the rough ER. So the ribosome and the rough ER work together to produce proteins, which are then packaged into vesicles. So these vesicles will travel to the Golgi body, which is a series of membranes that are kind of stacked together. And the Golgi body will help sort, modify, and repackage proteins into a new kind of vesicle. This new vesicle is going to have several possible destinations. It can leave the cell and secrete the protein if you need it immediately. You can store the protein in the cytoplasm for later use. You can become part of the cell membrane. Right? So the cell membrane is made up of the phospholipids, a little bit of cholesterol, and as you will see later on today, it also has quite a bit of proteins in it. And finally, it can become this other organelle called the lysosome. Okay, so lysosome, Golgi body are also part of the endomembrane system, right? The lysosome is responsible for breaking things down. It could break down macromolecules. It can break down worn out organelles. If the cell ingested something like a bacterium or a food particle, the lysosome will also be able to break those down. Okay, inside the lysosome is a mixture of enzymes, and sometimes if the cell is too worn out, if it is no longer functioning properly, then the lysosome can release all of its enzymes to trigger what we call apoptosis, right? We wrote it down last class. APOPTOSIS, apoptosis. That is programmed cell death. It's a killing off a cell deliberately for the benefit of the organism. Okay? And that's where we left off last time. Does anybody have any question before I continue with the lesson? Great. So the part of the endomembrane system that we left out was the smooth ER. We know what it is. It's a continuation of the ER with no ribosomes on them, but we haven't talked about what it does exactly yet. So let's go back to the PowerPoint slides here. Smooth ER has no ribosomes attached to them, and the main job is to produce different types of lipids. So rough ER makes protein, smooth ER makes lipid. right? Different types of lipids. There are phospholipids, there are triglyceride, steroids. All these are examples of lipids. They are made in the smooth ER. Depending on the function of the cell, sometimes the cell might have more of one kind of organelles compared to another. So let's say we look at the cells of the ovaries and the testes. These are the reproductive organs, right? That's where you will make the sex hormones, the testosterone, the progesterone, the estrogen. These are steroid hormone, as you recall from lecture 2. And so you would expect to have more smooth ER in these cells. Okay, something like a heart cell is not going to need as many smooth ER because it's not going to be secreting any steroid hormones, right? So the cell function is going to dictate how much of a certain organelle is going to be present inside the cell. Another function of the smooth ER is to break down fat and help detoxify drugs. So if you are on some kind of medication, antibiotics, for example, then you need to break them down. You need to metabolize them. That would be a function that takes place in the liver. So when you are on the drug, the liver cell can increase the amount of smooth ER as well as the detoxifying enzymes that are associated with it to cope with the presence of the drug. And once you finish the drug, you no longer need as many smooth ER, and the liver cell can, take away some of those. Right? So not only does the amount of organelle changes depending on the cell's function, the cell can increase or decrease on demand in response to the current needs of the body as well. All right, so that's a smooth ER here with no dots. That's the rough ER. Right? Rough ER is for protein synthesis. Smooth ER is for lipid synthesis, as well as detoxification of drugs. We did the Golgi apparatus already, lysosome we did already. And that's it. Okay, here's a quick concept check question. Ribosomes are made up of, I will just tell you the answer to save time because we do have a lot to cover. RNA and protein. Excuse me. Which of the following organelles are involved in breaking down macromolecules, toxins, and waste? Select all correct answer. So that would be the job of lysosome. It has various enzymes in it to break down macromolecules. The smooth ER, we just learned a slide ago, that it plays a role in detoxification. So those are your correct answers. All right. Now, after the first quiz, right, you should probably be aware that some of these questions actually does show up on the quiz. So make sure you spend some time to review them, for your upcoming quiz. Now we're gonna spend time talking about the other organelles. By the way, I am gonna give you guys a quick survey at the end of class. I'll post a link. Just 3 questions. It's anonymous. It would ask you just some feedback about the course, so that way I can improve your learning experience, right? So please complete that. If you have concerns about the quiz, if you have concerns about the way I teach, or if you have suggestions, feel free to let me know. All right. Any quick comments about the quiz though? Like, did you guys find it reasonable, or like it was completely, not what you expected? Any quick comments that you want to share with the class? No, everybody's happy, yeah?
I felt it was tricky, like some of the multiple choice answers, because
Mhm.
it's like so close to each other. So you really have to distinguish between what you're choosing.
Yeah, that's true, right? Not every questions are very straightforward, right? Because of the nature of the online testing, right? We cannot have questions that are all just very factual. Um, because then, people have access to the notes, right? So if it's all factual, then it's not really testing your ability to find information, right? That's not what we're trying to do. We're trying to see if you can analyze the information and pick out the right answer, right? Justine says it's harder than he thought. Um, yeah, well, hopefully, after seeing the first one, you kind of know what to expect, and hopefully you'll be able to prepare a little bit better accordingly. But like I said, right, make use of the tutoring service that's available. Ask questions, send me emails, right? Those are um, important in improving your grades. But did you guys find time an issue for some of you? Anybody feel like they were running out of time, or yeah, some yes, some no. So these are things you can let me know, right? In the survey. Again, is anonymous. So you can just tell the truth. Like you're not gonna make me upset. Like, okay, just just tell me what you think. If a majority of you think, time is not enough, I am flexible in increasing the time. All right. Like, I mean, I can't give you like, hour and a half. That would be, not appropriate for something that's this for quiz, but I can play around with the time a little bit. All right, so I'll post the link to the survey after class.
Just one
Mm-hmm.
question. Sorry.
Sure.
What was the answer for the first question you asked, the ribosomes? I missed that
Oh,
one.
that is the ribosome is made up of RNA as well as proteins.
Okay, thank you.
Yep. All right, now we're going to shift our focus to looking at other organelles. All right, the ones we've looked at so far, they are part of the endomembrane system, and now we have a bunch of other ones that we need to go through. The first one is a peroxisome. A peroxisome. They look very similar to the lysosome. Like, this is an actual picture of a cell, and there you see a peroxisome and you see a lysosome. There's no way for you to be able to tell. Right? There's no way for me to be able to tell other than, one is darker, one is lighter, right? So I'm not going to ask you to label something like this on the quiz. What I will do is show you cartoon pictures of the organelles, and you have to tell me uh, what they are, and perhaps tell me their function as well. Okay, so please look at the ones in the study guide. If you're able to identify them, you should be okay. So these peroxisomes are small round organelles, and they carry out oxidation reaction. Okay, oxidation reactions are reactions that involve removing electrons from molecules, and in removing electrons, you break them down essentially. One of the byproducts of oxidation reaction is H2O2. Okay, H2O2 is hydrogen peroxide. Okay, hydrogen peroxide is very toxic to the cell. That's why you need to contain these reactions inside the peroxisome. It's also responsible for breaking down fatty acids, amino acids, and a little bit of detoxification as well. So you can see the organelles functions overlap a little bit, right? So smooth ER, it does a little bit of detoxification for medication, for drugs. But then if we're talking about things like alcohol that needs to be broken down, then we need to have the peroxisome. Okay. If you track the movement of a protein produced in the rough ER, it can end up in all of the following locations except, okay, so please take a look. Okay, most of you got it right. It is inside a peroxism. That's not gonna be where it's going. All right. This essentially is asking you about this picture that we drew. Okay, like where is this vesicle going to go? Right? It can leave the cell. It can be stored in the cytoplasm. It becomes part of cell membrane, or it becomes a lysosome. Okay, it's not going to be going to the peroxisome. Okay. Now we're going to draw more pictures. I hope drawing the pictures actually helps you a little bit. You can let me know in the survey, right? Some people find it helpful to draw, others not so much. But I'm gonna keep drawing. Right? So next thing we're gonna draw is a mitochondria. Okay, that's singular. That's one, and plural is DRIA, mitochondria. Okay. So this is the powerhouse of a cell. Okay. It produces ATP. Using cellular, I guess we can say from, right? From cellular respiration. Okay. Now you might remember cellular respiration from lecture 2, or you might not, but that's okay. It is glucose plus oxygen, and that's going to produce carbon dioxide plus water and energy, right? So there are different kinds of energy, right? Like you think about the different electronics in your home, right? They run on different types of batteries, different cell types. Sometimes you plug it into the wall, other times you insert your AAA, right? There are different kinds of energy. So the preferred form of energy, the usable form of energy in a cell is ATP. Right? This is usable form of energy in a cell. Okay. We'll talk more about this in lecture 5. So let's take a look at the structure of the mitochondria. Knowing the structure is going to be important for later on in the course when we discuss the process of cellular respiration. You kind of have to know where things are happening and so on and so forth. So this is the mitochondria, the oval. It has an outer membrane. So out here is like the cytoplasm, right? There is the outer membrane. It's the similar composition as the plasma membrane. It's going to be made up of phospholipids. It's going to have a little bit of cholesterol, maybe a little bit of proteins in it as well. If there's an outer membrane, you can probably guess there is also an inner membrane, right? So the inner membrane is all folded up. It's all folded up into these finger-like projections, these accordion-looking projections. Inner membrane. Okay, so like this is the membrane, right? It's not smooth. The inner membrane is like, it folds up like this, right? Just like up and down forms these wrinkles, these foldings. Folds into finger-like just means it looks like finger, okay? Finger-like projections called cristae. to maximize surface area. Or cellular respiration. Cellular respiration is a chemical reaction, and you probably learned in your chemistry class that the more surface area you have available, the faster the reaction can happen in most cases, right? That's why like it takes longer to dissolve a cube of sugar compared to a spoon of sugar, right? The cube has less surface area compared to the granulated sugar. So by folding it up, by folding up the inner membrane, I should say, you increase the available surface area for performing the chemical reaction, basically. Now, there is a little bit of a space between the outer and the inner membrane. That is called the intermembrane space. Okay, we'll learn what that does later on. And in the innermost region, that is called the matrix. Okay. So again, the mitochondria is the powerhouse. It will allow this reaction to basically take place, and then it will start to pump out lots and lots of ATP. Okay. For every glucose that you burn, you will release up to 36 ATP. We need ATPs for everything. Okay, ATP is like money for the cell. Right? You need to pay for various things. You need to have ATP for movement. You need to have ATP to build molecules. Like if you try to build a protein, if you try to connect the amino acids together, that takes energy. That takes ATP. If you want to move things in and out of the cell, sometimes that takes ATP. Right? If you want to perform a chemical reaction, sometimes that also takes ATP. So there are various uses of ATP. All right. That's it. So what organ, what kind of cells do you expect to have a lot of mitochondria in it? What do you think?
Like protein cells?
Protein is a molecule though, right? It's not a kind of cell. But good try, Mina. Anyone else? Which part of the body use a lot of energy? Yeah, muscle, right? Justine said you move your muscle a lot, right? Okay, you run around, you hang on to bars and whatnot, right? Then they need to have a lot of energy. You would expect them to have a lot of mitochondria. Does that make sense? Remember, the function of the cell dictates what kind of organelles it's going to have an abundance of, right? Have a lot of, right? All right, I hope that makes sense. We will draw more later. This is the picture of an actual mitochondria. People asked me this morning, like, is it actually purple and blue? No, these are called pseudo color. Pseudo. Pseudo. Pseudo color. That means they're not real. Okay, pseudo means fake. Right? So this, like, if you look at a cell under a microscope, everything is like grayscale. Okay, they are actually no color as well, transparent, because they're so thin, right? Okay, but we can use dyeing techniques to color some of these things, and sometimes it's like you color them in post, like post-production, I guess you can, like after you take the picture, you can use software to highlight different parts of the structures of the cell of the organelles. Okay, so they're not actually purple, but that's what it is. You see the outer membrane here. We see the cristae, right? And that's how you draw it as a 3D picture. But we have all the necessary information in our diagram. Okay, so you can read through these things. It's exactly what I told you. You can read through them to reinforce the learning. All right, next we have something called there's something called chloroplast. Okay, chloroplast is found in plant cells, but not in animal cells. Okay, so our cells, human cells, don't have chloroplast. Plant cells do. You should know that in the world, there is not just plant cells or animal cells. Okay, there are other cells, right? Like there are like 5 different kingdoms, right, in the world. There is a plant kingdom, the animal kingdom. Anybody know another kingdom that exists, maybe? No, just plants and animals. Well, there are like fungus, right? That's another kingdom. And then there is like the kingdom for bacteria, right? And then there is another kingdom called protist. Okay, so some other cells, e.g., protist, also can have. They don't need to have. They can have chloroplast. We already learned 2 types of protists. There is the amoeba I showed you before, and then there is the paramecium. Okay, so they usually live in fresh water, and some of them can have chloroplast. Chloroplast contains chlorophyll. Okay, chlorophyll is green. It makes the plant green. If the part of the plant is green, it has chlorophyll. Okay, and the function of it is to carry out photosynthesis. Okay, so chlorophyll is green, and it allows the plant to capture sunlight. Okay, using the sunlight is going to carry out photosynthesis. Now, a lot of people have this misconception. They think the plant use photosynthesis to make energy, but that's not true. Okay, photosynthesis is a process that the plant uses to make food. All right, so photosynthesis is carbon dioxide plus water produces oxygen and glucose, right, with the help of sunlight. Right? And that is the food for the plant. Of course, the plant was stored as starch in either their stems or root, and then they could eat it, right? They could use that in cellular respiration. Plant cells also have mitochondria. They're able to burn the food to produce ATP for their own use. We cannot make our own food. We have to eat the food, and once we eat the food, we can use it in cellular respiration. Right? The chloroplast is also a double-layer membrane organelle, just like the mitochondria. It has an outer and an inner membrane. And then there are a bunch of stuff that's inside. We don't have to know them. Okay, so we can just cross out this last point. Okay, cross them out. So we good with the chloroplast? All right, so your next quiz number 2, is going to happen after Thanksgiving. I think it's going to be on the October 10th, if I'm not mistaken. 10th or 11th, I don't, I think it's the 10th, yeah. Yep, it's the 10th, right? And there will be 2 short answer questions, just like last time. And one of them is going to be on this slide. All right. So if you need to hear this part again, it's the 30-minute mark of the lecture recording. All right. I told you guys what's on the short answer question last time as well, but some of you still either didn't answer or like, you know, not answering it properly. So make sure you pay extra attention this time. One of the short answer questions is based on this. So this slide shows the relationship between the chloroplast and the mitochondria. Right? their functions basically complement each other, right? They are the reactants, the ingredients for chloroplasts are carbon dioxide and water. And that turns out to be the products of cellular respiration. Okay. Similarly, the reactants for the mitochondria for the cellular respiration are the products of photosynthesis, right? So in that way, they complement each other. Okay. So let me say that one more time. The reactants are the ingredients, right? The reactants of the chloroplasts, which are carbon dioxide and water, are the products of cellular respiration. And then vice versa. The reactants of cellular respirations are the products of photosynthesis. Okay? See, the world can have only plants left and no humans, and they would be fine, right? Because they have both chloroplasts and mitochondria. They can keep on making their own food as long as there is light, and then they will be able to use that food that they make to create ATP to create energy, right? For animals, not just humans, but animals in general, we don't have the chloroplast. We only have the mitochondria, and without plants, we wouldn't have any food source, right? Sure, you'd be like, we can eat other animals, but remember, if you follow the food chain, right, the animals that you eat, ultimately, there would be one that relies on eating plants, right? Like if you eat beef, for example, the cow needs to eat the grass. So without the grass, there's no cows, and then you will run out of cows to eat, right? Okay. So they are interdependent. These two reactions, one complement the other. Let's take a look at other structures that are found in plant cells and not animal cells. We have something called the central vacuole. The central vacuole is really big here. This whole thing is the central vacuole. It takes up, over 80% of the volume of a cell in some cases. And it's filled with water as well as nutrients. Right? Some vacuoles are found in the flowers of plants, and in those places, they would be storing pigments. Pigments is what gives color to the flowers, right? Some plants are poisonous, and they would store the poison in the central vacuole as well. Okay, so we don't have central vacuole. We don't have chloroplast. These are plant-specific structure. I have a chart coming up at the end that shows you what's specific to plants and what's specific to animal cells. This next one is an other kind of vacuole. It's not found in plant cells. It's found in some single cell organism. So this, you might remember this picture. This is a paramecium. It's in freshwater, ponds and streams and whatnot. And it belongs to the kingdom Protista, or it's a protist. Okay, so again, there's animals, there's plant, there's protists, and a few other ones. And one of the challenges of being a unicellular organism in open water is that the water keeps coming in. Right? It's going to fill up the cell, and if you don't do anything about it, it's going to get too big and it's going to pop, right, and die. So that's why the paramecium has this thing right here. It's called a contractile vacuole. It's basically a pump. Right? So it takes the excess water that's coming in, and then it pumps it out of the cell. Okay, it pumps the water out. And in doing so, it prevents it from swelling up and bursting. Okay. Another structure that is found in plant cells is called cell wall. We learned about cell wall before, right? It's made up of cellulose, a type of polysaccharide. It's very tough, very rigid, but it's not selectively permeable. Okay, like it just lets stuff pass through with no regulation, and it protects the plant cells from bursting. Okay. So human cells do not have cell walls, but something like a bacteria can have cell wall. Fungus can have cell wall. Fungus is not plants, right? You know that? Fungus, like mushrooms, right? That kind of stuff. That's not a plant because they don't do photosynthesis, right? If you try to grow mushroom with just sunlight, it's gonna die. Right? Mushroom, they feed off decaying matter, right? That's what makes them fungus. Okay, so they have cell wall as well. All right. Select all the false statement below. Go ahead, take a look. I will launch the poll for you in a bit. All right, hopefully finish reading. I'm gonna relaunch the poll for you. Okay, not a lot of participation today. That's okay. So let's see, the first one. Smooth ER is part of the endomembrane system. That is true. Okay, the endomembrane system includes nuclear envelope, rough and smooth ER, Golgi body, and lysosome. Okay, so that's true. We're looking for the false statement. There is an abundance of mitochondria. Let me just change that because it was wrong in the last class as well. There is an abundance of mitochondria in muscle cells. I think it was Justine who mentioned that. You know, muscle cell needs to move a lot. So of course you're gonna have a lot of mitochondria in them, right? So that's a true statement. We're looking for the false ones. All cells have cell wall to protect itself from the environment. That's not true. Human cells, for example, do not have cell walls. Hydrogen peroxides are found in not lysosome, right? It found in peroxisome. Okay, so that is false. Chlorophylls are found in chloroplasts. That's right. Right? Chlorophylls are the sunlight capturing compound. Right? You can buy chlorophyll online to add to your smoothies or something like this, like chlorophyll extract. But there's no reason to do that. It doesn't have any health benefit of consuming chlorophyll. They will tell you you will have benefits, but like actually not that I know of. All right, like you can just get your chlorophyll by eating your regular vegetables. Right? Eat your spinach, something that's dark green has a lot of chlorophylls in it, right? There's no need for extra chlorophyll. Doesn't do anything. Okay, that's it. Any questions before I move on to the last part here?
Just please, just a clarification. For when we're doing the vacuoles, the contractile vacuoles, you said that is not found in plants, right?
Yeah,
Just
not
the
in, that's right. Not in plants, not in animals, just the protist.
Just the protist. Okay,
Yeah.
thank you.
Yeah. Are humans part of the animal kingdom? Yes. Right. Contrary to what most people think, we are animals. Right. Figuratively speaking, and I guess actually speaking as well. But like like we are part of the animal kingdom because we we have the features of other animals, right? Basically. Okay. Last part. we got the cytoskeleton coming up. Cytoskeleton. The cytoskeleton refers to a network of fibers, proteins that are found throughout the cytoplasm. Okay, and their job as a whole is to either provide support for structure or helps moves things around or helps something to move. All right. So we have three types here: microfilament, microtubules, and intermediate filaments. Okay, so microfilament, microtubules, and intermediate filaments. Let's take a look at what they do. Does cell wall and cell membrane mean the same thing? No. Okay, all cells have cell membrane. Animal cells, plant cells, bacteria. We all have cell membrane. That's one of the features of cells. Cell wall is an extra layer that's outside of the cell membrane to protect the cell from bursting. Okay? So plants, they have cell wall. Our cells do not have cell walls. Okay. If you drink too much water, as we will learn later on, our cell will swell up and burst. Okay, so drinking too much water can actually kill you. If you water your plant, right, they love it. Right? You put a lot of water in it, they just stand up, right? Because they don't burst because they have the cell wall. Does that answer your question, Mina?
Yes. So you said the cell wall is just an extra layer from the cell.
Outside of the cell membrane, yeah.
Okay.
And it's only found in plant cells, or not humans, right?
Yeah, just in plants. Thank you.
Yep. So let's take a look at these three things. I know the names are confusing, okay? But that's why I encourage you to have the help sheet, right? I hope you guys did that for the first quiz. The first one is microfilament. It's made up of two entwined actin protein strand. Basically, you have each of these circles represents a type of protein called actin, and then you have a long strand of it. Okay, so imagine having two strands and then you just twist it up. Then you have your microfilaments. All right. When we look at a cell under the microscope, we can use different techniques to highlight different parts of the polyactin, very good. To highlight different parts of the cell. So in this particular picture, the person chose to highlight two features. Okay, to highlight the red circle right here in the middle. I hope you know that's the nucleus. I'd be slightly disappointed if you didn't know. That's the nucleus. And all these green strands that are all over the cytoplasm, that's the actin filament. Okay, that's the microfilament, right? Actin filament, microfilament means the same thing for us, okay? Like that, the actin makes up the microfilament. That's basically what it is, right? It's found throughout the cell, and it provides this scaffold for various organelles to attach onto. All right, so like it's the mitochondria, it's not randomly placed in the cytoplasm. They don't just like slosh back and forth, right? Like there has to be some kind of network, like a spider web, if you would, for things to attach on, to get stuck on. So there's the mitochondria, there's the mitochondria. If you have the endoplasmic reticulum, if you have the Golgi body, right, all these things needs to have something to attach onto, right? Otherwise, they're just like floating around, like it could get, displaced, right? It could get moved around too easily, right? So the actin, the microfilament provides attachment sites for various organelles. It also allows the cell to move. Now, this part is really hard for me to show on the slide here, but I'll try my best. Okay, so look, this is a cell, and then it has some actin here, and then some actin here, right? Now, let's say the cell wants to move this way. Cell needs to move around in the body. They don't, like, some cells don't go anywhere. They stay in one place, like your brain cells, right? But some cells needs to move around in the body, like your white cells, to catch bacteria, right? So if the cell wants to move this way, what it needs to do is it actually needs to break down all the actin at the back, okay? And then it will shove those actin forward. And what that will do is that will allow the cell to like extend like this, okay, with all the actin. And then it will grab onto a new point and pull the rest of it forward, right? And then now you would have inched forward. Do you understand what I'm trying to show you? Like, think of the hand, right? The hand is the cell, okay? Like, it wants to move forward, so it pushes all the actin forward, it grabs onto the new point, and then drag itself forward. Okay, pull, drag, pull, drag, right? That's kind of what happens. So the actin, the microfilament, allows things to attach in the cytoplasm, allows the cell to move around in the body. All right. Next, we have the intermediate filaments. Okay? Think of them as staplers. Right? There are places in the body where you would have two cells. Okay, one cell here, another cell right next to it. And you want to staple them together so things don't leak between them. Can you think of a place in the body where you don't want things to be leaking out? What structure? You want to keep the content in place so it's not going to leak all over the place. Anybody? Okay, skin is a good one, right? We don't want things to leak out from our skin. We certainly don't want things to, like, randomly leak into the body either. But anything else?
Your stomach?
That's right? Your stomach, your intestine, all those things have like food content in it, and you just don't want them to be like leaking all over the place, right? So that's why you need to have these intermediate filaments to staple the cells together. Okay, think of your intestine, right? Like, it's like a tube, and then you have the food inside. You don't want them to leak out. Right? Same thing with your blood vessels. You don't want the blood to be leaking out, right? So you form these intercellular junctions. Okay, staples the cells together. Also, the nuclear envelope needs to be pretty stable as well. And there is a type of intermediate filaments called lamins. Okay, and that allows the nuclear envelope to be stable so that it can protect the genetic material inside. People who have a mutation in the lamin A gene, the instruction to make this particular protein, have nuclear instability, which could cause the chromosome structure to fall apart, right, and interfere with gene expression. And that creates a condition called progeria. Okay? Some of you might have seen pictures like this where the person looks really old, but actually they are, 14 years old in this case, right? So progeria is basically accelerated aging. The person is still young biologically, but then it exhibits all the features of an old person, right? And that's because their nuclear envelope is destabilized, and it affects the stability of chromosome structure and gene expression. Okay, these people, there's no cure for this condition. There's actually a movie that was made about this condition. Right? I'm sure it's on YouTube somewhere if you're interested. Now, using gene editing tools, people are trying to experiment on, fixing some of these hereditary conditions, right? So if you know where the mistake is in the DNA, the idea is to fix the mistake, right, using gene editing. So there has been some promising results in mice to rescue a particular form of progeria. So that's a good start. But in reality, a lot of the success that we see in mouse model, they don't actually translate into humans. Okay, so we'll wait and see how this goes. This was like about 2 years ago. I have not read anything knew about it yet. So, maybe one day they'll be able to fix something like that. Okay, the last one is the microtubules. Okay, so we had the microfilaments, which is the spider web, right? The green stuff to attach things on to help the cell move. We just talked about the intermediate filaments for stability, for the staplers gluing cells together. And then the third kind is called the microtubules. Right? So I want you to think back to like an earlier slides. You don't have to flip back, okay? You can just listen. You know that vesicle that came off from the ER, right? And it wants to go to the Golgi body. How does it know where the Golgi body is? Like, what if the Golgi body is like all the way across the cell, right? Like, how does it know to get there, right? If we wait for it to slowly diffuse over, right, to move there by chance, excuse me, then it would take forever, right? So what happens is, it turns out there are these tracks, okay? You can think of them as like conveyor belts that are formed by the microtubules. Okay, so on the tracks, there are these special protein molecules called kinesin. Okay, and I kid you not, they actually look like this. They look like a stick person with 2 feet and 2 hands, but no head. They will grab onto the vesicle and they literally will walk on the microtubules and carry the vesicle to where it needs to go. You can observe this under the microscope. Okay, it's actually what's happening. All right, so it will take the vesicle from the ER, and then it will walk over all the way to the Golgi body, and then it will put it down, right? So the microtubules allows things to move in a more meaningful direction, right? It's not randomized, right? It's one of the characteristics of living things, right? Nothing is random. Things are very deliberately designed, right? This is a picture of a growing nerve endings, right? So how does the neuron knows where to grow and what connections to make? Well, in some cases, the guidance is the growth is guided by the microtubules. The yellow stuff here is the microtubules, and this is the end of a neuron connection, right? Trying to grow longer and connect with another neuron. Okay, so let's take a closer look at other examples of microtubules. There is another organelle called the centrioles. Okay, centrioles. Centrioles are yellow. I mean, they're not yellow, but they're usually yellow in cartoons, in pictures. They look like dry pasta, and you have 2 of them. They are found in animal cells, but not plant cells. The centrioles are going to be producing a special kind of microtubules called the spindle fibers. Okay, so the spindle fibers will attach to the chromosomes and move them around during cell division. Right? I already told you last class, right? The chromosome needs to go to different places when the cell is dividing. You need to make sure that one cell receives half of the genetic material and the other receives the other half. And how does it know where to go? Well, again, it's going to be guided by these microtubules called spindle fibers. Another place you will find microtubules are cilia and flagella. Cilia, you saw this earlier, it's found in airway, it's found in the fallopian tube. Generally, the cilia will cover the entire cell. Okay, the paramecium that I showed you earlier, they have cilia around the body that helps them swim around. For flagellum, it's just going to be one tail. Okay, one flagellum, usually. And sometimes 2, but like for sperm, for humans, only 1 tail. If a sperm has 2 tails, it doesn't know how to swim. It just like goes around in a circle. You can find videos of it on YouTube that shows you that. Okay, so these structures, they are made up of microtubules as well. This is just the cilia in the fallopian tube to show you. Right, so and the fallopian tube connects the ovary with the uterus, right? So when the egg comes out, the cilia is gonna brush it along so it knows which way to go. So this chart summarizes some of the unique stuff that are in animals but not plant cells, and then vice versa. So animals have lysosomes for breaking down macromolecules that are no longer needed, breaking down worn out organelles. They could trigger apoptosis. Centrioles, the pasta looking thing, right? That sends out spindle fibers for moving chromosomes. Flagella is, the tail of a sperm. Plant cells have chloroplasts to capture sunlight for performing photosynthesis. Central vacuole are really big, right? They store water and nutrients. Sometimes they can store pigments or poison. And then there's the cell wall, which is made up of cellulose. This is an additional layer that's outside of the cell membrane. It protects the cell from bursting, from lysis. Okay, L-Y-S-I-S. Lysis is to break. All right. Which of the following is made up of two intertwined actin protein strands? Do you remember? It's the one with the two red strands. What do we call that guy? Okay, I get my usual 7 or 8 responses. Very good. The correct answer is, of course, the microfilament, right? Yeah, this one. Oops. Macrofilaments. These are all microtubules, by the way, right? Like microtubules include flagellum is made up of microtubules, cilia is made up of microtubules, spindle fiber is made up of microtubules as well. All right. So that is it for lecture 3. Your quiz, like I said, is gonna happen later on after Thanksgiving, and it will be all of lecture 3 as well as all of lecture 4. Next week we will have the lab. I mean, you can start reading over the lab if you want. I think I might explain the lab a little bit on Thursday's class, and then on Tuesday next week we will I'll go through some of the more specific aspect of the lab with you to go through the actual assignment with you, and then you will have 2 days to finish it. You will submit it on in Thursday. Okay. So let's take a break. We will continue at 1:35, and we'll start on lecture 4. Okay, so please get those ready if you haven't already printed them out. See you in a bit. Let's move on to lecture 4. Before we do lecture 4, the PowerPoint, let's start with more drawings, okay? So please take out a piece of paper. If this is your paper, please turn it sideways. Okay, we're gonna be drawing a bunch of stuff here. It's probably gonna take up like half a page, like that. So this is called the fluid mosaic model. Fluid mosaic model of a cell membrane. So fluid means is dynamic. Okay, it's not static, right? So like the wall behind me, that's not fluid. That's like it's not moving anywhere, right? So cell membrane is not like that. A cell membrane it's fluid. It's able to change. It's dynamic. And mosaic means it has different components to it. Okay, so like think of a pizza. A pizza is mosaic. You can see the mushroom. You can see the pepper, the pepperoni, right? Different components of it, the pineapples, right? So it's not one thing. Right? In chemistry, you learn about homogeneous solution, right? That's one uniform phase. Cell membrane is not like that. It has different parts to it, different components to it. So that's why it's called mosaic model. So here's a cell. Here's the nucleus, and we are basically taking a slice of the membrane and looking at the structures that are associated with it. Okay, so this is what I want you to draw. Let's draw the phospholipid bilayer first. Okay, so phospholipid bilayer has a hydrophilic head, and then it has hydrophobic tails. Okay, two tails, right? Now I want you to put some that have a kink in it. Okay, not too many, just like, one or two here and there. Throw in a kink. And then here's the second layer. All right, and then leave a gap. And then draw more. Right, throw in a few kinky ones here and there. Have another gap. All right, that should be good. And you can put one, like a few more here if you can. Okay. So this is called the phospholipid bilayer. Hydrophilic heads facing outside, and then the hydrophobic tails are sandwiched between, right? The percent of saturated and unsaturated fat depends on the cell environment. I'll explain more later. Some environments will need you to have more saturated fat. Others will need you to have less of it. It depends. This is the inside of the cell. That's the cytoplasm. The inside. This is the outside. We call that the extracellular fluid, ECF. That's outside. Hopefully you're with me so far. Now, these gaps, right? That's where the proteins are going to go. So some proteins are going to go through the entire phospholipid bilayer. Okay. Some proteins are just going to be attached on either the inside or the outside. Okay. So that's called an integral protein. Goes all the way through. This is a peripheral protein. It's either on the outside only or on the inside. It doesn't go all the way through. The best way to think about this is think of raisin bread. Okay, think of a bread, with raisins in it. Some raisin you can see it on both sides of the bread. Okay, it went all the way through. Those would be like the integral proteins. Other ones are just stuck on one side, right? Those would be like the peripheral proteins. Okay. Now I want you to understand that this is a 3D structure, right? You don't have to draw this part. It's gonna make your diagram very messy, but I want to show you that, you know, this is a 3-dimensional structure. So you're gonna have like more heads, right? All over the place. Do you understand that? Yeah, let me just erase all these things. Right? So it's like this. Try to visualize this. You have a ball pool, pit balls. I think that's what they're called, right? It's like that. Like you fill up a bathtub with water and then you dump a bunch of pit balls in it, right? So like that's like a hydrophilic head. Hydrophilic head. They're just like covering the entire bathtub, right? So that's the situation here. Now keep that picture in your mind, all right, and see if this makes sense to you. Those balls are not static, right? They're not fixed in place, right? Like they could shift from one place to the other, right? And that's what fluid means. So like this phospholipid, it's free to move left and right. Okay, it's free to move horizontally, right? Like this one is free to move left and right. Okay, so phospholipid are able to move laterally, sideways. Okay. Sometimes, rarely, right? They can flip to the other layer. Okay. Like this one, like really, really rarely can flip over to the other side. Most of the time, it just stays within its layer. It's just moving sideways. Okay, it doesn't really flip. Okay, so that's what the fluid means. I hope you're still with me. Please ask me any questions. If you have anything, just ask. Now, some of these proteins, just type it in the chat box, okay? Or unmute and ask. Some proteins have these sugar on them. Okay, these are like glucose maybe. Right? And that makes it a glycoprotein. Okay, which are protein with sugar on them. I'll tell you what that does later on. Some of them, instead of sugar, some of them have lipids on them. Okay, so that will make them lipoproteins. Okay. Protein plus lipid. Okay. Again, I'll tell you what they do in a bit. All right. Now these proteins needs to attach to something. Otherwise, like the whole membrane is just gonna tear off easily. So you're gonna have these blue lines here. We learned this earlier today. Can you tell me what those are? Please tell me you know what they are.
The filament thing?
That's right, the filament thing. That's right. So these are the actin filament, right? Okay. Actin filament. Right? It allows the proteins to attach onto it. Think of the membrane as like a tarp, right? Like if you go you know, picnic or something, you put at the beach or something like that, you put the tarp down, and then like you want to anchor them, right? Into the sand, into the soil, right? Otherwise, when the wind comes, the whole thing's gonna flip, right? Same thing with the cell membrane. You need to anchor them. And that's what these actin filaments are for. Okay. All right. One last thing that we're gonna add to this amazing picture. We're gonna have these molecules that are inserted between the phospholipids. And those are the cholesterol. Okay. The function of cholesterol is to help maintain fluidity. Okay. More cholesterol equals more liquid. Less cholesterol equals more solid. All right. So keep that in mind. We're going to come back to this in a bit. That's it. That's the fluid mosaic model. Does anyone have any question about what we've drawn?
Yes.
Yeah, go ahead.
Are the proteins, so like the integral proteins, is it attached to the phospholipids?
Yeah,
Like
I wouldn't say they're attached to it, but like, you know, they fit snugly into it, right? Like the phospholipids kind of like create like a hole, and then it just sits right in it, right?
like a pocket, like it's
Yeah,
stuck,
exactly, right,
like
exactly.
a pocket. Okay.
Again, as a loose analogy, right? Like, you know, think of a sandwich, and then you take a carrot stick and then you poke right through. Carrot stick would be like the protein. Okay. Like, is it attached to the bread? Not really, but like it's like snugly, right? Is cholesterol a type of lipid? Yes. Yeah, so there are two types of lipids in the cell membrane. There's the phospholipid that makes up majority of it, and then there is the cholesterol, which helps adjust the fluidity. You don't want the membrane to be too fluid; everything would just leak out. You don't want it to be too solid either. Nothing passes through in that case, right? You want it to be just the right consistency, and by changing the percent of cholesterol, you can adjust the consistency. Great questions. Any more? Okay, so I'll post this afterwards. If you think of anything else, you know where to find me. Let's go to our PowerPoint slides now. Okay. So this lecture is about membrane transport. It's about how things get in and out of the cell using various methods. But before we get to that part, we have to understand the structure of the cell membrane, right? We just kind of went through quite a bit of that. Let's take a look at what the slide says. So that's the professional drawing. We basically draw on everything, right? So part of what I want to achieve with the drawing is for you to build the structure from scratch that will help you understand it a little bit more, rather than, you know, me just show you this picture and, you know, you will likely get overwhelmed, right? And now your job is to take a look at what we have drawn, compare to the stuff here, and you know, just match them up, right? So for example, there's the glycoprotein that I talk about. There's something called glycolipid, which we haven't drawn, but that's okay. You will still understand it. Glyco means sugar, lipid means, well, lipid means lipid, right? So these are lipids that have sugar stuck on them, right? Okay. There is the integral protein. There is the erythroprotein, so on and so forth. This tells you what each one of them does. With the exception of glycoproteins and glycolipids, I've told you what everything else do already. So, you know, we'll come back to this glycoprotein, glycolipid thing in a bit. But for now, you know, we know what the cholesterol does. It maintains the right fluidity. This makes up a majority of the cell membrane, right? Now, this is another star question, okay? This is the second short answer question on the quiz number 2. What's the first question about? Were you guys paying attention? Can someone remind me? What was the first question that you have to know for the quiz number 2, for the written response? That's right, about the chloroplast and the mitochondria and how they're related, right? Okay, so if you need to refresh your memory, remember, watch the recording, skip to 30 minutes. If you need to hear about this thing again, then skip to hour and 20 minutes, I would say, roughly. So here, the question says, how would the percent of saturated, unsaturated fat and cholesterol content of a cell membrane found in plants growing in cold environment differ from those growing in hot environment? Okay, so let me write this out for you. Okay. So this is cold environment. This is hot environment. Okay. So we're gonna look at adaptation needed. We're gonna look at percent of saturated fat and percent of unsaturated, as well as cholesterol. Okay, so I'll give you a moment to draw the chart, and then I will explain. All right. If it's really cold, if the environment is really cold, do you expect the cell membrane to be more solid or more liquid? Okay, think about leaving butter in the fridge. Is it going to become more solid or more liquid?
Solid.
Right, more solid, right? I hope the rest of you agree. Now, we don't want the cell membrane to be too solid, because if that's the case, then nothing goes through, right? So you want to counteract that. You want to undo that, right? So cold environment tends to make cell membrane more solid, right? So the adaptation needs to counteract that, needs to oppose that, undo that, right? Which means we need to do things that will make it more fluid. It's very important for you to understand the difference between this and this part here. If you're confused, ask me a question. Like I am not sure what you're confused about, right? So like you need to tell me so I can clarify for you. Okay. So again, if it's really cold, cell membrane tends to become solid. You want to undo that. Okay, you want to have features that will make it less solid. Right? Think of it like this, right? Like if it's minus 40 outside, you can't use your regular windshield fluid, right? You spray it on, it's gonna freeze up. We don't want that. So we need to add anti-freeze to it to make it not freeze. Right? So basically it's the same thing. It's so cold, the cell membrane is freezing up a little bit. It's a little bit solid. So we need to change it to make it more fluid, less solid. So what do you think? Should we have more saturated fat or less saturated fat if we want it to be more fluid? More saturated fat or less saturated fat? What do you think?
Less.
Less.
That's right. We want to have less saturated fat. Very good. What about unsaturated fat? More or less if we need it to be more fluid?
More.
More, right? We learned this in lecture 2. This one we learned today, cholesterol. If we want it to be more fluid, do we want more cholesterol or less cholesterol?
More.
More. Yeah, I hear 3 more. That's right. Right? I told you. More cholesterol, more fluid, right? That's it. So that's what you would expect. And that actually is what happens for plants that are growing in like cold environment, like in the alpines or something like that. If you look at their cell membrane, they would have less % of saturated fat. More of it will be unsaturated, and more of it, there will be a little bit more cholesterol in it. On a hot environment, it's different. Hot environment tends to make cell membrane more liquid, right? Because it's like melting a little bit, right? So you don't want it to melt. You want to do things to make it less fluid, right? More solid. Okay, so what do? Well, it's the opposite. You need to have more saturated fat, less of the other two. Okay. You shouldn't be memorizing this. You should be trying to understand it. All right. So that you can, apply it on the test, right? Any questions before I move on? All right, so now you know what the next 2 questions are gonna be for your quiz number 2. So we know there are proteins in the cell membrane. Some are integral protein that goes all the way through. Others are peripheral proteins that just attach on either the outside or the inside. We want to know what those proteins are there for, right? There are 6 functions associated with these membrane proteins. Let's go through all of them, and then that will be the end for today's class. Okay, so this just refresh your memory on what integral proteins are. They go all the way through. A peripheral protein might be attaching on the phospholipids, or sometimes they attach onto the integral protein. It's like a little buddy, okay? There's the integral protein, and then a peripheral protein just, stick onto it. First function, I already told you this one, right? They provide attachment sites to the cytoskeleton. Right? In our picture here, you see that's the actin filament. They got to attach to something. And that's what the membrane proteins are for. They provide anchorage points to attach. Not just to the inside. Like, don't do this to your picture. But on the outside, you also have cytoskeleton. Okay? These are called the extracellular matrix. It's just fibers on the outside, right? So the cell needs to attach to those as well, and that's what the membrane proteins are for. Okay, some cells have very unique cell shape that are related to their function, and to maintain those cell shape, to maintain those cell shape, you would need to have anchorage point from the proteins. Okay. Number 2, cell signaling. The body cells communicate with each other through hormones in most cases. Okay, so we know there are protein hormones, there are steroid hormones, and in some cases, the protein hormones or the hormones will attach to a membrane protein and it will cause the cell to change its behavior. After all, that's what hormones are for, right? So let's say you finish a meal, you release the insulin, which is a hormone, then the insulin will bind to your body cells and tell them to absorb the glucose from the food, right, from the bloodstream, right? So that changes the cell's behavior. Without the hormone, the cells would not know what to do with the glucose, right? So here is another example. We have epinephrine here. Some of you know it by the other name, adrenaline. They mean the exact same thing. It will bind to a receptor protein, which is on the surface of the cell, and then will cause a bunch of reactions to happen. There's a fancy word for these reactions called signal transduction pathway. Don't worry too much about that name, but it's just a bunch of reactions, right? Which ultimately causes the hydrolysis of glycogen, okay? So you will need adrenaline for running away from danger, or like, in this case, preparing for a race. And that is energy demanding. So it makes sense for the cell to perform hydrolysis to break down the glycogen so you get more glucose. All right. Now the protein the hormone rather, never got into the cell, right? It just binds to the receptor protein and then it causes all these changes inside the cell. This reminds me of Halloween for my family as I was growing up. My parents didn't like Halloween. Okay, something about handing out free candies, they don't like that. Okay, so every Halloween, if someone rings the doorbell, my mom would be like, hide in the basement, pretend no one's home. Okay, so this somehow feels like the situation, right? The epinephrine is like the kid. This is the doorbell. They ring the bell. They never came into the cell. They never came into the house. But then it causes all these commotion, all these reactions that are going on inside the cell, all these changes, right? And that's what the receptor protein is for, okay? It relays the information from the molecule and causes all these reactions, all these changes inside the cell. That's how your neuron function as well. Okay, we'll learn more about this next semester, but when a neuron needs to communicate with another neuron, they do so by releasing these neurotransmitters. You might have heard of things like dopamine, serotonin, right? You might have heard of antidepressant called Prozac. All these will behave like some kind of neurotransmitters, and then they will bind to a receptor protein and causes the next neuron to behave differently. Okay, so this receptor is a membrane protein. Okay, so first function is provide attachment sites. Second function is to function as a receptor to relay the signal from a hormone. Next is to perform enzymatic activity. This is not new. You already know that proteins are enzymes sometimes, and they help speed up chemical reactions. But in some cases, you need to perform multiple reactions, one after the other. Okay, so you need to take this thing, and then the first enzyme will convert it to another thing, and then a second enzyme will convert that into a third thing, and so on and so forth. So there are 4 reactions here that requires 4 different enzymes. Rather than having the enzymes randomly scattered throughout the cytoplasm, then it would take very long for the ingredient to find the next enzyme, right? So rather than doing that, you just put them all in the same place, okay? One after the other. So you can perform the chemical reactions very rapidly, right next to each other, okay? So this is the third function of membrane proteins. Next one is intercellular junction. We already talked about this a little bit earlier with the intermediate filaments, right? I said you want to staple adjacent cells together so things are not going to leak past them. And you guys said, skin is like that, right? Intestine, blood vessels, they're all like that. So it turns out the intermediate filament is not enough. The staplers are good, but you need more reinforcement. And that's where you would need to have more membrane proteins, right? So, you have one membrane proteins in one cell, another membrane protein on the other, and you just kind of glue them together, right? So it works together with the intermediate filaments to form tight junctions. That's what they're called. Next is what we call cell-cell recognition. So the glycoprotein, the sugary protein, and the glycolipids, and the lipoproteins in some cases, all these things, they collectively form an identification tag. Okay, identification tag, an ID tag, almost like a barcode for the cell. Everybody has a unique set of these identification tags. Okay, your cell is going to be different than my cells. No one on this planet is going to have the exact same set of ID on your cells unless you have an identical twin. Okay, that's why if people need to have an organ transplant, you cannot just randomly take the liver from one person and give it to another, right? Your immune system would recognize the foreign organ as having a different set of ID tags, and then they will attack it and you will reject the organ, right? Okay, so that's why you have to do matching between donors and recipients. And even if you find a very good match, right, it's still not 100%. So that's why people who are transplant recipient, what do they have to take to prevent rejection? Does anybody know? No? They have to take immunosuppressants, right? Drug that suppresses the immune system so that they don't reject the organ. Okay. Anybody knows their blood type here? Do you know your blood type? Do you want to share them with the class? You can make it up. I would never know you're lying. Really? Nobody knows their blood type? I find it very surprising.
A positive.
Okay, yeah, with some A positive, O positive, 2 O positive.
Negative.
Negative, okay, and then a third O. Yeah, very good, right? Some of you are A, some of you are B. I'm AB positive, right? So when you talk about your blood type, let's say you're type A, it turns out you have glycoprotein A on your red cells. If you are type B blood, you have glycoprotein B, right? So these gives your cells identities, right? Okay, depending on what kind of proteins you have on your red cells, it determines whether you can transfuse to other people or receive blood from other people, right? So that's what cell-cell recognition means. Okay, allows your immune system to distinguish what belongs to the body and what does not. Self versus non-self. Here is a white cell that is eating up some kind of foreign bacteria. That's not supposed to, not bacteria, some kind of foreign cell here. That's not supposed to be in the body because it has a different ID tag. Last but not least, the membrane proteins are important in transporting materials across the cell membrane. That's going to be something we'll go into a lot more details next class, but whether something can come into the cell or not depends on what kind of molecule it is, and sometimes it's really big. And if it's really big, you would need to have the help of a plasma protein, a membrane protein rather, to help move it over. Okay, and that's transport. All right, just 2 poll questions and we will be done. I will do this one with you. According to the fluid mosaic model of cell membrane, which of the following is a true statement about membrane phospholipids? First one, they are free to depart from membrane and dissolve in the surrounding solution. That's not true. Okay, what this is saying is this. They are saying, one of these things, one of these phospholipids, maybe they don't like it to be there, and then they just walk away. They just go into the surrounding and dissolve, right? Of course, that's not going to happen because if it did, your whole cell membrane is going to basically dissolve and fall apart, right? Okay, so that's not true. They occur in an uninterrupted bilayer with membrane protein restricted to the surface of the membrane. That's not true. They are not restricted to the surface of the membrane. As you saw, some goes right through, some are found on the inside, right? So not just on the surface. They have hydrophilic tails in the interior of the membrane. That's not true. The tails are hydrophobic. They can move laterally along the plane of the membrane. That's true. Remember those balls, the ball balls. They can move sideways, right? Okay, but they do not flip. They don't flip-flop. Right? So they frequently flip-flop. That's not true. All right, so this is one of the harder questions. And like I said, right, like I don't know if you noticed, but like some of these questions actually shows up on your quiz, right? So make sure you go through them when you're studying. Okay, last question. Which of the following is not a function of membrane proteins? We just learned 6 functions. Attachment to cytoskeleton, cell signaling, enzymatic activity, cell-cell recognition, forming intercellular junction, like gluing 2 cells together, and transport, right? Number 6. So if you compare it to what I said with the list, right? This is not a function of membrane proteins. It doesn't protect you. It doesn't protect the cell from lysis. Lysis means breaking, right? Burst, right? And that's it for today. All right, we will do the rest next class. And again, please take a moment to fill in the survey. It's only 3 questions. It will give me an idea of how you guys are doing in the course, and if there are any like requests of anything that you want to see changed in the course. All right, enjoy the rest of your day. I will see you all on Thursday. Okay, thank you. Bye-bye.